And Dr. Didar also won the 2023 McMaster Faculty of Engineering Entrepreneurship Award. And in 2024, he received the University Scholar Award. And he has over 20 granted or pending patents and has presented over 60 national and international invited talks and keynote speeches in Asia, Europe, and North America. So with that introduction, the floor is yours, Dr. Didar.
Thank you very much. I appreciate this. I know Ali for such a long time, and it's been an honor. I know him always as a mentor, and it's great to be here at Terasaki and then give this talk. I'll tell you briefly where we are. As you see in the map, we are somewhere between Toronto and Niagara Falls in Hamilton. If you've gone from Toronto to Niagara Falls, you've probably seen us and you're passed through us. And as you know, the Canadian side is much better than the American side. We're also, this is our campus at McMaster in Fall, which is quite beautiful. And we're also the city of waterfalls if you want to visit. We also take credit for Niagara Falls as well, although it's a little bit far from us. So today I will talk to you about biointerfaces. I will try to tell you some stories of biointerface-based materials we have developed, and we've tried to commercialize some of those, and I'm going to give you some challenges and advancements we have made in these. If you look into a variety of medical devices, such as indwelling medical devices, drug delivery systems, tissue engineering, lab on a chip, organs on chips, you always see an interaction happening between synthetic material and biological material. And in that case, proper design of this interface is the key for successful implementation of that device. If we put some certain imaging devices aside, such as MRI and other instruments, most of the biomedical devices have this interface, and its optimization is quite important. I'll try to tell you in this slide what I think are the most basic elements of a biointerface, and you might want to even help me complete this slide as I try myself as we move forward for the past couple of years. So normally we have a complex biological sample. This could be blood, urine, a tissue in the body, and then sometimes we have a target that we want to interact with. Sometimes we want to detect this target, but sometimes we want our implant or biomaterial to interact with this target. And we have a specific ligands for this purpose. I always tell my students that we're in engineering, and to find these targets, I put an antibody here, but it could be, aptamers, peptides, many different types of biomolecules. We go to the people in the biology department and ask what kind of specific markers we can use for this target. But other than that, we always have an interface that we immobilize these biomolecules onto them. And when we immobilize them, it's important if they're physically immobilized or covalently attached. Their orientation also is very important because they have a specific binding sites, and that also determines the success of that interface. And by the way, this interface doesn't have to be a flat surface. It could be a nanoparticle, a microparticle, a fiber, you name it, a well plate, a microfluidic device, an organ on a chip device, and so on and so forth. And then we need to properly block this surface, especially if this is a sensor. The proper blocking of it is very important for preventing non-specific adhesion and noise down the road. And then we need to bring this target in contact with this capture molecule, right? This could happen with different ways. If we're doing an in vitro assay, this could be through diffusion mainly in the lab. But if we want to add a little bit more enhancement to this, we could use, the diffusion is there, but we could use microfluidics that brings convection. Or in the body, we could have situations where there's flow, and that could help enhance that interaction with that target that we have. And for microfluidics, we need to know obviously about flow control and fabrication. And at the end, if this is supposed to be a biosensor, then we need a transducer that converts that signal that we have actually captured to that target into a signal that we can actually notice or see and receive. And if you, and there are many different types of transducers, obviously, I'm sure you already know about this. But now if you look into a variety of biomedical devices, such as biosensors, labs on chips, organs on chips, implants, you name it, all of them have either all aspects of these or some of them. For example, a catheter needs to have proper blocking so it prevents coagulation on it. But if it's supposed to sense something, then it needs to have a molecule that senses and then a transducer that tells you what's going on. And recently, there's a lot more increase on in vivo sensors as well. And then you need kind of a wireless communication system as well that sends you the message. Let's say if you put a sensor on an implant, so that implant not only needs to interact with the tissues around it to integrate to it, but can also, send you signals of an infection or other markers inside the body to outside. And these are the elements of a biointerface. And I think all of us working on biomedical devices, we're dealing with aspects of this slide a lot in our research. And then from now on, I'm going to tell you some stories of what have been involved and what have developed on these biointerfaces. The first one is the work I did when I was at the Wyss Institute on lubricant-infused surfaces, or they also are called the SLIPS. Slippery liquid-infused porous surfaces. This was initially developed in Joan Eisenberg's lab at Harvard, and it was inspired from pitcher plants. So if I can play this video, let me just try to turn off my... so if I can play this video, in a dry day, insects can easily walk on a pitcher plant. But as soon as it rains, the nanostructure together with the wax that the pitcher plant produces, it turns it into an ice ring. And insects can easily fall into it, and it's a really nice meal for the pitcher plant as well. So inspiration came from this, that to develop the initial SLIPS versions. And then later on, we developed a smoother version of these surfaces that are used for biomedical applications, for tubings and well plates and other types of applications. As you can see here on the left side, the video shows an acrylic that's coated with this coating, and the right one is the one that's not coated, and you see what happens when we put the blood onto these surfaces. The principle is very simple. So you put molecular brushes onto these. Here are fluorinated ones, but now you can put silica-based brushes, and then you use a compatible lubricant on top of that that's matched with that chemistry on the surface. And this blue is the lubricant that creates a physical gap between the solid surface and whatever comes as a biological liquid on top of it. And that prevents, for example, blood attachment, coagulation, bacteria attachment, and biofilm formation. It basically gives it the self-cleaning properties. So when I started my lab in 2016 at McMaster, I developed a variety of versions of these lubricant-infused coatings. We applied them on medical switchers and surgical meshes. As you can see at the top, these are switchers and meshes that are commercially available, and we've induced clotting on them, the beautiful clot formation you see on the top, and then at the bottom are the ones that we have treated that significantly prevents coagulation on them. And later on, a PhD student of mine, Mariam, who is a prof at Calgary now, developed catheters based of this. And as you can see in the video, the catheters that she developed is actually quite slippery, and the blood doesn't want to interact on those compared to the others. Then we treated pipette tips with this to prevent cross-contamination. And then Matthew developed this coating on oxygen sensors for wastewater. When you put oxygen sensors in wastewater, it falls significantly because it's full of bacteria. But when we did the coating, the sensors were able to go for a month without losing sensitivity, which in the other case, they always, even the commercial ones, fail within days. And this was quite helpful to prevent that. We use it for microfluidic molds to make the surfaces smoother when they come out of cheap 3D printed devices. And we also made extruded liquid infused surfaces that are entirely liquid infused bulk material. And as you can see in these videos, that how they behave when droplets are exposed to them. So this is quite interesting. We still continue to work on these surfaces, but then I was talking to Jeff Weitz, who is a hematologist here, a great colleague and friend here at McMaster, and he told me these are great, but I'm interested to apply these on vascular grafts. But if you wanted to do this on vascular grafts, we don't want just to repel everything. We want to recruit endothelial cells to the site of the graft because they need to form an endothelium layer. So normally for a surface, if you, let's say, use a chemistry like Apt's chemistry with EDC NHS, you can put antibodies on it. But when the blood comes on it, it attaches there, forms a clot, and basically messes up the entire system. If you use lubricant-infused coating, it repels everything. So you're not able to recruit these red cells, which are endothelial cells in this case. But if we use biofunctional lubricant-infused surfaces, then we're able to specifically capture the targets while repelling the others. And the way that I explain this is, imagine there's a shallow sea with a meter of water on it, and there are trees sticking out of this. So that C is the microns thickness level lubricant onto the surface. And the trees are these antibodies or any capture molecule you want to put there. And whatever you bring there as blood cells or whatever, they're like boats that are floating onto this shallow sea. And then when the target is there, that tree basically captures that boat. When it's not there, it lets it go, and then at the same time prevents coagulation and biofilm formation and so on. So we applied these on a different grafts, vascular grafts, starting with PTFE-based grafts that are often used in the clinic, and we showed that the ones that we functionalized with CD34, which is a epithelium, which is a endothelial cell, progenitor endothelial cell marker, we're able to recruit endothelial cells on these while preventing coagulation from happening, and compared to commercially available ones that are used on the patients. You might ask, so how patients are taking these? Because they are prescribed with anticoagulants. Most patients who have vascular grafts, they need to take warfarin, for example, for the rest of their lives, which increases a lot of chances of bleeding and other side effects. So the goal of this project was to implement this sort of biomaterials that decreases the use of anticoagulants in patients who go through such surgeries to enhance their, outcome after the surgery. We applied them onto Dacron grafts as well. So Dacron grafts are different. They're like fibers. On the left side, you see a commercially available graft where we've induced clotting on it. But on the middle is the lubricant-infused one. The right one is the biofunctional lubricant-infused one, where we have the capture molecules on it. And as you can see, it significantly suppresses coagulation while attaching endothelial cells onto the surface. Then we did all of these experiments in static conditions. They were not under flow, but as you know, blood in the body is under flow, and it has a certain shear stress as well. So we thought we should design an instrument that we can test these in vitro, inducing physiologically relevant shear stress. And that's what Veronica Butt did, who is a PhD at Stanford now. And she developed what we call the vascular graft on a chip. It's a very simple microfluidic device that enables you put any type of biomaterial embedded inside. So we don't functionalize the channels. We actually embed the actual biomaterial into the chip. And it could be a variety of different, we've tried many different biomaterials with this: nylon, PTFE, PMMA, and so on, and PDMS. The one that I'm showing here is mainly PTFE, because we wanted to evaluate the vascular graft's effects under shear stress. As you can see on these images, when we put the vascular grafts under this device and induced physiologically relevant shear stress, they showed also significantly suppressing the coagulation while promoting endothelialization onto the surface. Some surgeons pre-endothelialize vascular grafts, and that could be done here as well. So we could actually seed cells on them before putting them onto the chip and testing them. And the designs is somehow that you can vary the shear stress along the channels as well to see where you have the highest effect of either inducing thrombosis or tissue integration. So these were quite interesting, and we thought that, okay, we have these functional lubricant-infused coatings, and why not we use them for biosensing? Well, a major drawback for biosensing is non-specific binding, and that messes up the signal and background noise and sensitivity of the sensor. So we started applying these for some applications in biosensing. First, Amit, who's a postdoc with the Radisic lab at UFT now, made these well plate-based assays for IL-6 detection. So this is what I'm talking as an islands here. So we have these capture antibodies, the rest is lubricant, and then we put target there. In this case, we're using blood for detection of IL-6, which is a cytokine, and it's used in a lot of infectious disease and even cancer-related as a marker for the progression of the disease. And what Amit showed that, oops, sorry, going too fast. So what Amit showed that we can create these microarray-based well plates and then put them into the format of a well plate, adding a superstructure in it. And this could be used to detect IL-6 as low as 0.5 picogram per milliliter, which is pretty good for a fluorescent-based detection system and within the clinical levels for this. Then we applied this for detecting E. coli in milk, as you can see in the middle, but I'm going to talk about that as we get to the food project. And later on, we applied this for electrochemical detection of clinical samples, mainly UTI in urine samples and also septic patients. And we were curious to know if we apply this lubricant-infused coating, can we actually have any charge transfer and use it for electrochemical detection? But we designed the system in a way that we were able to get the signal. So these are conductive surfaces that are also lubricant infused, and we can basically use them for detection of certain cases. And as you can see here, we're using it for urine and blood samples, which are quite complex samples for biosensing. And this enables us to do this within an hour for a urinary tract infection. And as you can see, the clinical data there, we were able to identify all the positive E. coli samples that came from Hamilton Health Sciences compared to the healthy patients. The same with the septic patients. So this was all interesting, the lubricant-infused coatings. At some point, I started thinking that this is all great, but what if we expand the application of these surfaces for high-touch areas, such as doorknobs and subways and airplanes and so on, so that they prevent attachment of pathogens and their spread? The idea seemed interesting, but we couldn't obviously use lubricant onto these surfaces. First of all, it becomes slippery. The lubricant for an extended period of time could evaporate. And then we started thinking of other ways to create these nanostructure surfaces that are pathogen repellent. And then I'm going to talk about Fendex, which is the startup that came out of this work. And since we're a public company, I need to show you this slide. So you already know that we have a lot of high-touch surfaces. We always worry, it's a flu season now, you go to the airport, hospitals, places that people touch the surface. And there's always a chance that you're going to touch one of these surfaces if you're sick, and then someone else will do, and then they're going to get sick. And then we thought of lotus leaf in this case. I'm sure a lot of you have heard about the lotus leaf. So lotus leaf has these nanostructures on them that gives it self-cleaning properties. The opposite of the lubricant-infused coatings, lotus leaf maximizes air pockets on the surface, creating a Cassia-Baxter condition where it gives it a super hydrophobic conditions with over 150, sometimes 160 degrees contact angle onto these surfaces. A lot of people have tried to do lotus leaf inspired surfaces. One of the biggest problems with those is that it's very hard to scale them up for commercial applications. So you might be able to make these in clean room, but when it comes to making millions of them that could go on a lot of doorknobs and touch surfaces and so on and so forth, it's really hard to scale them up. So we thought of these shrink films. These are olefin polyurethane shrink films that are normally used in packaging. And we developed a process that we create 3 levels of hierarchy on these films. One hierarchy comes from, in microscale, comes from heat shrinking and wrinkling them. The second hierarchy comes from nanoparticles infused on them. And the third one is a molecular level using basically an intact or inert molecular level onto the surface. So I don't want to go through details. If you have any questions at the end, we can talk about those. But this is a commercial sarin wrap at home. When when the food is extra, you don't want to waste it. You wrap it around put it back in the fridge. This is the same exact wrap as we have treated with this specific coating. It gives us repellent properties, and as a result, it repels pathogens significantly. And it can be scaled up. So we're actually working with a facility in Philadelphia now, and we're able to make these at hundreds of meters a minute. Or sorry, an hour. So it's quite scaled up. So we can go in an hour or so, we can make, very large amounts of these films. And we tested them with priority pathogens, drug-resistant ones, such as Pseudomonas and MRSA. You can see on this SEM images, the left side are the stained or colored SEM images with bacteria on a normal wrap, and the right side you see the ones on our repel wrap. We also tested them with some real applications such as a stethoscope and pen and key. Excuse me. And we also showed that if you touch these surfaces that are contaminated, our hand does not pick up these contaminated surfaces. So this was the history about this was that this was all before COVID. We published this, I remember after it was published in ACS Nano, CNN tweeted it, and we got a lot of attention on these services. And then COVID came, and a lot of people started reaching to us, and can we use them? Even a lot of hospitals started calling us, can we use these in our facilities? And as you remember, during COVID, everybody was very careful of surfaces. Later on, we found out that maybe aerosolized transmission is more important than surfaces. But then it became a very big motivation for us to try to commercialize this. So that's where we started Fendix in 2021. And we got we very significant amount of funding for Fendix. Fendix went public in last March in 2023, and it's a public company right now. And then we developed different versions of these pathogen repellent material for a variety of applications. One that we're also pursuing at Fendix is catheters. So we're creating medical catheters that prevent biofilm formation or coagulation. Either say, for example, in a urinary tract catheter, there's a very high chance of biofilm clogging, and they need to be changed often. So we see a really good path here for developing these sort of catheters. We also developed fluorine-free versions of these. As you know, there's a lot of concern about fluorine due to its environmental effects. And then we have patent-developed versions of these that are fluorine-free. And then later on, we developed spray versions of these. So it's not a film anymore, and we're quite excited about this. These are sprays that you can spray onto various surfaces. It could be during manufacturing, or somebody could apply this at home, and it gives the same hierarchical structure to the surface. But we've also added antimicrobial properties to this, such as silver and gold nanoparticles, and we're scaling this up as well currently. An interesting experiment here to observe, I guess, is this graph. We tried to contaminate surfaces with a finger that's been contaminated with MRSA. So we contaminated the glove with an MRSA solution, and then we kept touching surfaces. So as you can see here, we went up to 50, we stopped at 50. But if your hand is contaminated with the pathogen, potentially you could go and touch 50 different surfaces and contaminate all of them. And that's how a major route of transmission, specifically for bacteria in hospitals, or for flu viruses that are heavier, could sit on the surface and survive for a while. Well, when we used our spray, we showed that even after first touch, we have over 3 log reduction, but after a second touch, there's no pathogen being transmitted onto the other surface. And that shows how important these surfaces are in preventing the transmission. And then recently this year, we developed a newer version of these, and we call them smart fabrics. These are surfaces that not only repel and kill if a pathogen sits on them, but they report the presence of bacteria onto the surface. So it changes the color. So we've added some sensing capability to it, that if there's a certain threshold of bacteria sitting on it, it changes the color. For example, we thought of curtains at hospitals. You know, at hospitals, people come and clean a lot of different surfaces, but it's hard to clean the curtains. They might change the sheets, but the curtains remain, and they could get contaminated. So having a curtain that is repellent and antimicrobial, but at the same time changes the color if there's something growing on it, would be very interesting to focus and you know, could help. And we started using AI for a lot of these applications to kind of help distinguish between a contaminated surface versus a clean surface. So I'm going to talk a little bit about our smart food packaging material here. As you know, we waste tons of food. I guess in the US, over $200 billion worth of food is wasted every year. Same number in Canada per capita. Canada has actually more food waste per capita compared to US and UK as well. And I was actually in LA last month, and there I heard on the news that California government announced that they're getting rid of the best before dates on food packaging, which is I think it's great. Everybody should do that, and it also gives a lot of you know, motivation on what we're doing and what I'm going to present here. So we want to prevent contamination, alert consumers and producers if there's contamination, prevent massive recalls and waste of food, but at the same time try to extend the shelf life and also make sure that the food is used as much as it should be, because the expiration dates on the food packages are very conservative. And if you see milk or a meat expires today, it might actually be good to consume for the next 2-3 days. But we toss it out, it goes to landfills basically. And we see three pillars for this, three kind of approaches for us. One is detecting pathogens in food individually in packaging so that we prevent massive recalls. The second is detecting spoilage, which I think it's more important for preventing waste. And the other one is biocontrol, how we can actually control and extend the shelf life of food. I'm sure you all know about these, and I mentioned it, so I'm just going to pass on this slide on the importance of this and tell you what we have. So current methods for let's say detecting pathogens in food are very time consuming. Normally they open a packaging, they take it to a central laboratory, put it in a stomacher, juice it out, plate it overnight, and then it needs at least 24 hours to know if there's a pathogen in there or not. What we wanted to do is to create sensors that goes inside the packaging that can alert you if there's, you know, 4 or 5 major food pathogens there. To do this, we are using RNA cleaving fluorescent DNA enzymes, RFDs. So we call them DNA enzymes. These are DNA molecules with enzymatic capability that could trigger a signal if a certain food bacteria, pathogenic bacteria such as E. coli is present. And it doesn't need to be the E. coli itself, but also what E. coli secretes could trigger this signal. And these are discovered through a selects process, the DNA sequence. And then you could use a fluorescent-based approach or a colorimetric-based approach to kind of trigger the signal when the target is there. And we call these bioinks. So we kind of conjugate them with functional molecules so it could be printed onto the food packaging material. And this is what you see on the packaging material when you have this DNA sequence here. And then if the bacteria, the target bacteria is present there, it triggers a signal, in this case, a fluorescent signal, and you can pick up the difference and then say that, and you can use a handheld fluorescent device to kind of realize this and say that there's contamination. So initially we tested this with, you know, apple and meat and other food samples and showed that these flexible sensors could go inside the packaging. They're good for the shelf life of the food, and they trigger the signal. As you can see in this case, this is E. coli positive, this is negative, so it can tell you if the packaging is contaminated. Later on, we incorporated this into those lubricant-infused coatings for very complex environments such as milk. We call them lysozymes. And this enables, you can actually print these onto the milk bottle. And then if the target pathogen is there, it triggers a signal. You can scan it and find out if that's contaminated or not. Then after E. coli, we're looking into other pathogens. We finalized E. coli and Salmonella, as you see here. So for Salmonella, we did a different approach. We tried to target the RNAH2 enzyme coming from Salmonella typhimurium for the target. So we designed the probe against that. And then a lot of people were interested in colorimetric detection here. So we incorporated gold nanoparticles into the system and then designed a very simple sensor with pipette tips. So we conjugate gold nanoparticles onto the surface of the inner lumina of these pipette tips. And then when you basically take the Salmonella, let's say, is more common in chicken, if you take the juice coming off the chicken into this pipette tip, if it triggers a signal, you keep it there for 20 minutes to 1 hour. You dispense it down, and if there's Salmonella there, the color changes. Very similar to a rapid COVID test or a pregnancy test, and you can find out if Salmonella is present. There, I kind of compare this to a rapid COVID test compared to a PCR COVID test. And we validated the system in Germany with one of the large food testing companies. And they're very excited about this because it takes them 24 hours to culture these and find out. By using this could be done within an hour or two to find out if there's Salmonella, for example. And the same concept could be applied for E. coli or Listeria that we're currently working on. And then everybody started asking us, so if you put these sensors on the food, how do you know the pathogen is there? It might be somewhere else. How you're going to make sure you're capturing everything? Although we know a lot of the time they're on the surface of the meat, not inside it, but it could be still in different areas. So we redesigned the packaging. We opened the window under it, and then we included a membrane inside that design. The membrane, first of all, holds the reagents, food-friendly reagents that are necessary for detection, but also creates a pathway for the juice coming from the chicken, let's say, in a packaging that comes down to that membrane and enables the detection. So I won't go through the chemistry, but I just want to show you some of the results where we actually put a whole chicken, and we contaminate this with different ways. So we contaminate a knife or a hand, and then we make sure that contamination transfers to the chicken. And then we use this sensor to a handheld fluorescent device currently to detect this. It could down the road could be a colorimetric based detection as well. And this is I think this paper came out yesterday. And again, to move towards colorimetric based detection, we created these hydrogels made of DNA and DNA enzyme molecules, and we embedded color reporting nanoparticles, in this case gold nanoparticles. It could be other types of coloring agents inside it, and the gel is intact when the target is not there. When the target comes, triggers the gel, it breaks it down because it breaks down, it activates the DNA enzyme, breaks down the sequence, and then the gel is gone. So the E. coli, for example, is there. But another innovation here is that we used bacteriophages, viruses that are specific towards that target bacteria, and then it lyses the bacteria, but it lyses our target bacteria. It's not like any other lysing agent that could generically lyse any bacteria there. It lyses the target we're looking for, because as you know, phages are very specific towards the strain of the bacteria that we're trying to target. And as a result, the gel breaks down, and we know that the target is there. So we call it phage based amplification, a hydrogel that's basically uses phage based amplification to detect. And then we used AI to predict the results, in this case of if the gel is gone or not, for any challenge in terms of making a decision if the gel is still there or not, especially at the early stages. So we tested this with lake water samples from Ontario, showing that we can detect 10 CFUs of E. coli in lake water, as well as clinical urine samples coming from Hamilton Health Sciences. And in all cases, we were able to detect urinary tract infected patients using these gels, and hoping that down the road in the future, these could be simple devices at home that people could use to detect certain conditions instead of going for central laboratories for these. So this video gives a summary of what I mainly talked about. So we have contamination that happens in a lot of surfaces, especially in food. If we use certain types of repellent surfaces, they can prevent these in the first place from happening in samples. And then if we use these DNA enzyme molecules in the form of a bioink, as we call them a bioink, then this certain configuration can be immobilized onto the food packaging. And then if the target bacteria is there, it's going to trigger a signal, and we will know by that signal if that's present. This could be down the road multiplexed, and we could detect multiple types of pathogens, not only one. And these sort of sensors could be as barcodes that go inside the packaging down the road. And that's the vision I'm talking about, that we don't have best before dates anymore, but we scan the food for contamination. I'm not going to talk much about the spoilage. We have a couple of technologies in the pipeline for spoilage detection that could all be packed into that barcode that could tell you if there's, you know, some sort of a contamination there or not. And it's not just what you do in research to develop such technologies, especially in food. It's so complicated. So we work with a variety of institutes, companies, government agencies to bring all this, what we call the Sentinel group, together to move this forward. If you're interested about food packaging, I encourage you to look at these two papers. The one in Nature Reviews Bioengineering is about commercialization of food packaging material, and the other one is more about smart food packaging materials that came out recently. I'll spend a couple of more minutes on biocontrol, and I want to focus on bacteriophages. These are organic antimicrobials found in nature. They don't, you know, cause resistance. They don't create resistance, and they don't, you know, destroy your microbiome because they're very specific towards the target bacteria, right? So if you don't know about phage, phages are viruses that only infect bacteria. They are very specific towards their target bacteria. They attach to the bacteria, go inside, they take over the machinery of the bacteria, create hundreds of its copies, and then burst the bacteria, and then they can go after other bacteria. And they're quite diverse. There's probably a phage for any strain of bacteria, but it also has its own challenges for implementation. But because of all the challenges for antimicrobial resistance, as you can hear, every day we have new cases of resistance that are coming up. There's more and more interest towards using bacteriophages, both in food industry and for clinical use. Europe is quite advanced than North America on this. In a lot of places in Europe, for example, in Belgium, pharmacies can actually prescribe phage right now. You can just go get it from them. But what has remained unmet in food in phage is, you know, developing new types of materials that help enable its potential. So we have a couple of platforms for phage. One is we have developed these pure phage microgel sprays for food. Phage is actually approved for use in food applications, but then it's not effective as it could be. So these hydrogel-based pure phage particles that are packed with phage could actually have a much higher impact. And then I'll briefly mention to you about what we call phage tablets. These are dry tablets of phage that rapidly help with screening of phage, which is a major issue to find the target phage. And we are also developing implants that are incorporated with phage. And I'm not going to go through this, but we're getting really interesting survival results with sepsis models, for example, using these implants. The first one I mentioned to you are these bacteriophage biomaterials. And I do this in collaboration with Dr. Du's lab here at McMaster. She's the main expert in bacteriophage biotechnology. They had developed these gels initially, and they were going to test them in animal studies. Then COVID came, and the animal facilities got shut down at McMaster. And we thought of what should we do about this. And I suggested them to test this in food. And then we started testing these for food samples. And we showed that it can, you know, if you put E. coli 0157, a specific phage, it could actually eradicate them within a couple of hours in food samples. So this contributes to eradicating and biocontrol within food samples. The second option I mentioned is about scalability and transportability of phage. Normally, when a patient is at the hospital, the antibiotics don't work, there's no hope, they look for phage. It's really hard to find the phage. You need to go to a central laboratory and screen thousands of phages. And these are, old-fashioned ways of screening them that takes days, or at least a day, to find the target phage for it. We thought that we should be able to include all the biochemistry together with phage, or a cocktail of phage within dry tablets, that these could be easily added to the sample coming from the hospital and give us a yes or no hit within half an hour to 1 hour. So we know that which one of these could be the target for this. And that's what we did here. So we developed these phage tablets that are the concept is based on detecting ATP when the phage is killing the bacteria, but we put the ATP reagents together with the preservance sugars that preserve phage, and then we created a single tablet of this. So these tablets could be preserved, could be transported everywhere around the globe, and then when that clinical sample comes in, you just need to add that solution into the well plate that has these tablets on, and within half an hour to 1 hour, it actually signals to you if that phage is the match for that bacteria. So it could be prescribed for the patient. So we tested 4 different bacteria, 9 strains, and over 75 phages for this specific study. One example is that a patient comes with a urinary tract infection. It's resistant. It comes to the lab. We put these on these multiple type of E. coli phages we have. We already know it's E. coli. It's just resistant. And then we find the hit here. The P32 would be a hit, let's say, for this case. I believe this is a Pseudomonas one, not E. coli, if I mentioned E. coli. And you can see all the strains of we tested again for E. coli. So we tested multiple different phages. And then this could turn into a larger screening system where you can test against different types of bacteria. So we're hoping that these phage tablets could actually, help accelerate the use of phage for clinical applications and discovering the target phage for patients that have basically no hope for treating them because of antimicrobial resistance. So I hope I've been able to tell you why biointerfaces are important. We can do a lot with designing proper biointerfaces, either pathogen repellent surfaces, bioimplants, sensors. I didn't talk about our organs on chips and organoid systems, but in designing these systems also biointerfaces are important. And I think proper design of them would be the key for their success. I want to thank all the agencies, government, private sector who have supported us throughout this, and these are the champions who do the main work. And thank you again for inviting me.
All right, thank you very much, Dr. Didar, for that great presentation. I think it's a very good type of science that is also very useful, not only for diseases and health, but also I think food is a big part of also how humanity has to also manage things in the world. So there is a few questions that came online. So the first one is how is the accuracy and the repeatability of your biosensing? Is it CLSI mandated or CLSI M100 compliant? And if it repels the pathogen, does it kill them as well?
Yeah, so I introduced a couple of different sensors, so I don't know which one. So for example, for food, if I wanted to mention that we're doing with salmonella, let's say. So with salmonella, we have 10 to the 3 CFU for limit of detection per gram of food. So for food, they say per gram, not per milliliter when they're doing the detections. And for example, in Canada, I think it's the same in the US, the threshold for salmonella is 1 CFU per mil or per gram of food. So we're not in that threshold yet per se. But again, this is a rapid test. Most of the time, the ones that really make people sick have very high concentrations of these bacteria that are going to trigger the signal. Anyways, they're not going to be that low. But the government rule says that it needs to be as low as 1. But this could be a rapid test versus a PCR test, let's say, that can tell you rapidly how of this could be done. That's mainly for the food samples. We haven't incorporated it specifically for food killing as well as detection at the same time, although these are separate technologies in the pipeline. But I'm hoping at some point these could come together for that specific application. In terms of the standards, you said I didn't catch the standards, but we try to follow to make sure the repeatability of these, the signal to noise ratios, the CV values of detection, specifically when we're measuring microarray fluorescent intensity or colorimetric intensities, that we have significance and repeatability when it comes to these. So as we worked with so many industries on these, they have an eye on making sure that we basically follow those standards, that when the time comes, we can meet those.
All right. Well, good. Thank you for the answer. So another question is, can you shed a little bit more light on the bacteriophage production and how, uniformly you can produce them, and how are those, like, purification process really applicable for GMP production and so on?
Yeah, great question. So one of the reasons that phages might not have gotten the attention they should is purification, actually. So, well, first of all, antibiotics took over, but phages still is used in Eastern countries. In Eastern Europe, phage is quite popular, but they're found in nature. You can, isolate them, store them, and propagate them. So they're propagated by infecting bacteria and making more and more of them. But let's say if you infect E. coli with the phage for target for E. coli, when it lyses E. coli, you might have something like LPS. That's kind of an endotoxin that's present there. And if you can't purify it from the phage that you produce, it's going to cause a lot of issues down the road. But recently, the past, I would say 10 years, the purification technologies have advanced significantly, that right now, especially if you go to Europe, there's no challenge in purifying phage and making sure that it is GMP, GLP level, could be applied for, a clinical application. So that issue, which was a big bottleneck, I guess, for phage, has completely been resolved right now to purify.
All right, sounds good. Thank you. So another question is also regarding the efficiency of the biosensors. So if they lose their efficacy or efficiency upon degradation, or in the case of hydrogel, maybe upon swelling, what are the different strategy to really keep those packages durable? Because I guess for food, you can have some food that are kept for maybe a week, 2 weeks, and some have a shelf life of much longer times.
Yeah, so the reviewers actually made us test these. We did over a month of storage of these to check for swelling, and in different environments, different temperatures. So they're quite stable within that. I guess one issue when you develop such gels is that, they give you false positives. They dissolve on their own without even being triggered by a target. So we've resolved all of those. They're quite stable. I think we've tested for over a month, but I think they should be good for a very longer period of time to test these. And the good thing is that if you give it a bit of a time, it actually triggers, dissolves, and then you basically can even see it with the naked eye. That's what's happening.
Okay. So then after, maybe another question related to that is then, if we add those sensors to the packaging, how is it really going to influence the price tag of the final product for customers?
That's the fantastic question. That's $1 million question, right? And what recently we've turned more into spoilage. We're just developing AI apps for predicting spoilage and stuff. Main reason is because you go to meat producers, they tell you cook meat well, don't worry about it. They don't want to actually use these sensors because they see it as a liability. What if it turns out positive? But you bring food, like you bring meat at home, you put it on the counter. I don't know about other people, but at our house, kids come and touch those counters, and then they get sick. That's raw, meat, and then you need to know if there's bacteria in the meat, even if you think you cook well and it's gonna be well. And a lot of times you don't cook well, and then they're there, right? So, but they don't want to add to the price of the packaging. So although compared to other technologies that's out there, like electronics and other versions of technologies that are very expensive because they require more expensive sensors, these DNA enzymes, as soon as they're discovered, they're very easy to scale up. They cost pennies, but we still have issues even adding pennies to the cost of the packaging. And I think that's something that needs initiations like what California did for best before dates. that kind of forces the producers to move towards that direction. And I hope we have more of that happen.
And so in that sense of like where do you think those things can be implemented faster? So I know like you mentioned there's a big difference between Europe and North America, and after I don't know how it is in Asia or actually after in Africa and more like poorer country, but like where do you think this type of technology really is going to be deployed first, or who would adopt this type of technology?
Yeah, it's hard to say. I think at least we see more interest from Europe than North America.
Mm.
So far, a lot of people have contacted us, Portugal, UK, Germany. We've had a lot of Belgium interest coming from them. I know in East Asia, there's a lot of interest. For example, places like Japan, if you've been and you've seen, they're crazy about packaging. They want to add any interesting technology packaging. And interestingly, Toyota came and invested in our projects. So, and I think it also goes back to their roots as well, coming from Japan and their interest on developing sustainable and innovative technologies that could, be added there to make it better. So it's $1 million question where it's gonna start, but I feel more advancements are happening in Europe and East Asia than in North America.
After another question is like, do you think there is any limitation regarding a pathogen repellent surface treatment spray that might be a threat for the environment? And how how can you protect the environment if it's a threat?
Yeah, so it's a great question again. If you start something, you always need to evolve. If you start the technology, you generated iPhone 101, you need to go to iPhone 2, 3, and so forth and so forth. So, for example, the films we made, I remember somebody tweeted under the CNN tweet that you're going to wrap the world with plastics because these are films that we put, but it's an immediate, solution to a big problem. But then we thought of developing sprays of these so that they eliminate the need for plastics. And the other concern is using PFAS fluorine-based materials in these. And we've kind of resolved that moving out of fluorine and using other types of materials that are more efficient. But, even if you look into your screen, monitor, your phone, there is fluorine on these. So how do you think your phone is water repellent or your iWatch is water repellent? It's because of fluorine on it. So it's really hard to get rid of these. We hope we can do that as well. But at the end of the day, those are the challenges for environment that we need to look into in these technologies.
And so, and I know you haven't touched much on your organs on a chip or like those applications, I guess, for maybe some research and so on. And so, like, do you have maybe any insights from you using that technology with in vitro type of model and like seeing maybe if there is any changes or like influencing mutations or something like that in those like living system, I guess?
Yeah. So yeah, I mean, I'm coming from, my postdoc was in a lab that they're quite hardcore on organs on chips, right? And we started something on gut on a chip that still continues, but it didn't end up being a major drive force behind our lab. We do cell cultures. We do we work on micro nano robots and their interaction with cells or biofilms or validating them in organs on chips that other people and other co-ops are developing instead of using them for animal studies. But specifically for gut, we had the celiac disease model on a chip that we recently had a paper on that as well. And the reason we focused on celiac because we had a specific genetically modified mice model of that at McMaster that was quite unique. So we were able to develop that celiac disease model on a chip, a humanized version of that onto the chips. As you know, I'm sure Terasaki is quite focused and quite advanced in using organs on chips, but it doesn't leave much room for us to compete with such institutes. So we try to find those niche areas that we could actually make an impact.
Okay. And so then the follow-up question on that, because again, like you said, I also, I do organs on a chip and I'm very interested in, vascularized models. And so I was interested in your vascularized graft chip, where again, you try to some extent functionalize the endothelial cells using specific receptor. But, we know that like, especially angiogenesis or vascular remodeling, we know that, or lumen or blood vessels are to some extent a little fluid. So how does, making a cell stick to only one place and locked into place, how is this really affecting the remodeling or vascular remodeling that can happen in vitro, especially if we implant the device?
Yeah, no, it's a great question. So we don't make them stick into one place, so we don't pattern. We entirely coat the surface with, let's say, endothelial specific markers. But here's the trick. So the surgeon I work with, they keep telling us that you need a couple of days to prevent thrombosis happening on these when they're implanted. After that, the lubricant could be gone. And if you have those markers there, they could help endothelialization. So the ones we're doing with vascular grafts, let's say, it doesn't localize cells in certain areas. It actually creates tight junctions of endothelial cells in the entire surface, while at the beginning preventing initiation of clotting.
All right. Okay, sounds good. So then maybe the one last question that I had. So again, I think it's very interesting that you can repel to some extent microbes and other things. And so, do you think this type of technology could be used in vitro cell culture, where we create a sort of good-free type of things, where we can do, or antibiotic-free type of culture, or like, do you have any insights on this aspect?
Yeah, I mean, we're really trying, and I'm lucky that that we have this collaborator, Zainab here. We work very closely on phage and using phage as an antimicrobial, and I mean, we try to say it's a major issue, but it's actually becoming a major issue, and everybody's talking about, you take antibiotics, you kill all the bacteria in your body, and you need to eat a lot of probiotics to come back from that, right? So trying to implement phage targeted in certain areas, even, creating phage biomaterials that could be even applied locally, shear thinning hydrogels that could be applied into the site of an infection that has phage on them. These are, I think, things that could be done with phage that could really help with preventing infections and promoting or preventing resistance on antibiotics. The same with in vitro stuff. I hope when you're using your organs on chips and you're trying to challenge these with bacteria and using antibiotics, you also consider using phages with these.
Yeah, I think that's what the next step would be for sure. All right, well, with that, I think that was the last question. So I really want to thank you very much for your presentation. I think it's very insightful, and again, it shows also how to apply biomaterial engineering in many different area of humanity, I would say, not just only health and so on. So thank you very much, and have a very happy Thanksgiving and everything.
Oh yeah, thank you. Yeah, it's Thanksgiving in the US. I completely forgot about that. Happy Thanksgiving and enjoy. And thanks everyone for coming and joining. I appreciate it.
All right. Thank you very much. Yeah. Bye-bye. Thank you.